Good afternoon, everyone, and welcome to the first seminar of the Translation Manchester Research Network seminar series. Before we start, can I please ask you to keep your microphone muted and your camera off until the end of the talks. If you do have any questions, there will be a question and answer session at the end of each talk, each presentation, and you can also put any question at any time within the chat, and we'll pick them up at the end of each talk. My name is Alessandro, and together with Becky and Jim, we manage the Translational Partnership Award. This is a welcome-funded initiative that is aimed at facilitating translational research at the University of Manchester. Today, we're launching the Translation Manchester Research Network Seminar Series. This is a series of monthly events, which will feature the partners of our network, which is basically a collection of institutions and facilities that supports translational research in Greater Manchester, not only at the university. Presenting today, we have Scott from Affinity Biomarkers and Penny from NCITA. However, before we go into the main talks, I'd like to give you a brief of introduction of Translational Manchester and the network. If I can please also ask you to stay until the end, because we're going to launch a short feedback poll in order to understand if this meeting meets your expectations, so they can improve it going forward for the next events. So, as I mentioned before, Translation Manchester is funded by the Wellcome Trust. Our main goals and aims are listed in this slide. We want to support translational research in order to translate discoveries that are happening at the university within the healthcare setting, and we're doing that by providing funding to academics. We help them connecting with other academics across discipline, connecting them with clinicians within the NHS, connecting them with industry. Ultimately, we want to increase the way that academics are innovating with industry, and we want to generate that change in research culture so that innovation becomes at the core of what the university does and translation. So we have developed this translational pipeline. You might be familiar with it. We divide divide research in translatable to the left and translational to the right. We have 4 stages of translatable research. So this is discovery science. And then we have 4 stages of translational research. These are corresponding to the technology readiness levels. When we speak with academics, we try to identify where they sit within this translational pipeline. We try to understand what blocks them to go from one stage to the other. And ultimately, we try to use our expertise in order to unlock these bottlenecks and shift the research across these translational continuum. Obviously, we don't have all the expertise between myself and Becky, so we heavily rely on the Translation Manchester Research Network. This was funded in 2019 when Translation Manchester was first established. It now includes over 50 partners, and these are across the university, but Greater Manchester in general. It includes also companies, the university core facilities, all the biobanks, clinical trial support. And the idea is that as a translational research facilitator, are able to signpost academics towards the partners within the network that are able to support that translational research at any stage of translation. For that, we established this booklet that you can download at this link, and also Jim will be probably posting that in the chat as we speak. And the booklet has got information on all the partners and the type of support they can provide to the academics. And in order to engage better with the academic community, we're launching today this seminar series in which the partners of the network will be able to provide information to the academic community about the support that they're able to give, and at what stage of the translational pipeline it's best to contact them. It's best for the academic to contact them. A little bit of advertising here about our activity, Translation Manchester activity in the next few months. Obviously, the seminar series will continue. Next month, we have planned the next event on the 22nd of March. If you want to save the day, although communication will follow. We are delivering together with the BRC in the Innovator Training scheme starting in March and spanning until November. We are launching an early career researcher competition, providing to ECR up to £2,500 to access training, visit collaborators, all aimed at improving the the translational aspect of the research. We're also going to be holding a symposium. This is hopefully going to be face-to-face, although it looks like it might be still virtual in the summer or autumn. And it was going to be a follow-up to the launch event that we had back in 2019. Our funding call are going to be open again in the summer. We've got two schemes: Access to Expertise, HUE, and Project for Translation, P4T. The call, as I said, will be opening in the summer with the project awarded and started by November time. And we have more details about all of these activities. If we have time at the end, we can provide more info. Otherwise, you can just visit our website and information will be available there. So without further ado, I'd like to introduce our first speaker, Scott Harrison from Affinity Biomarkers Lab. And Scott will be talking about how Affinity Biomarker can facilitate translational medicine from lab to treatment. Scott got his PhD at the University of Auckland after getting his degree in Cardiff. He was a key member in funding a series of successful companies that include Plant Metabolomics, Metabolon, and also established several bioanalytical labs around the UK, US, New Zealand, and Denmark. And now he's Senior Director of Scientific Services of Affinity Biomarkers. Scott, the floor is yours.
Thank you for the presentation. When you go on your training classes, you always are told never apologize, but I'm going to apologize. This being the first talk in this series, I struggled knowing where to pitch it. So I took the opinion that I'd pitch at quite a low level, and if there's any questions or follow-up that people want, you're more than welcome to find my details from Translation, and we can have a chat. So I work for Affinity Biomarkers. Affinity formed out of the London Hospital University system, and we're a translational medicine company. Our aim is to actually take ideas from academia and SMEs and help them translate through those development pipelines that have just been highlighted. So we deal from D4 through to T4, that type of area. We were initially based in Imperial College in London, and we're planning on opening up a lab which should have been up and running by now. But of course, we ran into the problem that was COVID, and that put us huge delays on our expansion plans and moving in labs just for logistical purposes. We're involved throughout the UK, currently with multi-centered COVID trials, including biobanking, proteomics, and metabolomics of COVID and long COVID. So Affinity's aims are to assist, as I say, academics and SMEs and companies to validate their biomarkers and make sure that they can get them into the right place at the right cost. We support academia, biopharma, and with clinical trials, and our expertise is both in the analysis, but also in facilitating, pulling together the larger clinical trials that people need. Our mission, of course, is to make sure that you guys can get what you need when you need it, and at a price that is favorable, of course. So, of course, around that, we have to consider quality. We comply to the Human Tissue Act. We're in the process of 15189 ISO accreditation. We've got a quality management system. We track all the samples that we handle, and we have an alliance with Biocrates for doing targeted metabolomics on large scale. So the structure of Affinity is the clinical chemistry and immunoassays are based down at our new lab in London, and the mass spectrometry lab will be based up in the Manchester facility, and hopefully will be moving forward at a speed once the opening up from COVID occurs. So the Manchester mass spec lab will have standard assays for metabolites, for drugs, and we'll also be setting up a targeted and possibly a non-targeted metabolomics facility there to meet all the needs, all your needs from discovery through to validation. So that basically is the sales pitch over, and I decided, as I say, to pitch at quite a low level. And quite often when I meet with clients, I have pretty much the first meeting is the same conversation over and over again, and that's because basically we're divided by a common language. The meanings of certain words and certain implied understanding between biologists, clinicians, and analytical chemists, which I am, mean that we can, you can ask what you think is a relatively simple question, and I go away and give you a complicated answer, and I tell you that your analysis is going to cost a lot more than you think it should do. And the usual questions that I'm asked is how much of my compound is in this sample, or what is in this sample, and then the obvious question of how much will it cost to tell you that. The idea, if we look at the first question of how much compound is in the sample, we've got to pull out what we mean by how much. So to an analytical chemist, how much is a question which can be answered in multiple ways, from is there any of the compound in the sample at all, through to is there more of that compound in this sample versus a standard or a different sample. Do you want to know exactly? Do you run it with a calibration curve? Do you want more precision? Because of course, all the measurements we make have errors built in there, and I'll talk more about errors a little bit later. But to mitigate some of those errors, we can either use what are known as surrogates, which are compounds that are similar to the analytes, but structurally a little bit different, or we can use isotopically labeled standards. Of course, as we go down this list, the cost increases, the accuracy which increases, usually the time to develop the methods increase, and of course the cost increases, which is something that's important for you to remember, especially if you have thousands of samples you want to analyze. So, some common sources of errors: the biology itself in living systems, variability, especially in compounds that are non-homeostatically controlled, can vary up to 30%. There's collection and storage, both absorption, losses in sample storage, be it in a fridge or a freezer. One example I have of losses due to storage is I was running an assay that relied on measuring of acetate, and we, as was common practice, shoved the samples in the freezer over the weekend and reanalyzed some just to ensure that the levels were coming out the same, and lo and behold, they weren't. It turned out that freezing the samples pushed the acetate out of the solution, and of course, it then being volatile, it evaporated from the sample and was giving false readings. And it turned out that for storage of these samples with acetate, it was important to actually refrigerate the samples and subsequent testing showed that these could last for over a week in the refrigerator, whereas a simple freeze thaw would destroy the accuracy that we achieved for the samples from the freezer. There are systematic errors, miscalibration of pipettes, errors in the hardware that are repeatable, and then there are random errors, which are the user errors, the missing of an air bubble in a pipette, and so on and so forth. So we also would like to know what you want to know about your analyte. And chirality in biological systems can be very important. Molecules that have a reflection or a rotational symmetry can behave very differently in biological systems. So it might be important to you to know which enantiomer or stereoisomer you have in your system. If you want to know this, of course, it complicates the system. And the last thing your analytical chemist would want is to report back to you a bulk analytical result and it not be the result that you want. We also want to know about the physical properties in the case of acetate, the volatility, if the samples there that are stable, or the analytes are stable in the samples. If you want to analyze ATP, you've got to take special precautions and analyze them quite quickly. Will the sample ionize in the technique that, or it may dictate the technique that we have to use if the sample is not suitable. So for later I talk about HPLC or mass spectrometry. If it doesn't analyze, we might want to use HPLC. If it does ionize, we might want to use the mass spec. The other point, which is for the safety of the analyst, is the sample does it contain hazardous materials, which may be, of course, generally speaking, having worked in this field for too many years, we always treat everything as if it's hazardous, but it's always nice to know if there are any known hazards, including infectivity, such as from COVID or HIV or the like. So it's important that a assay should be fit for purpose. And the reason I put thin layer chromatography here is I had an assay where a biologist walked up to me and said, I want to know, is there any, how much of a compound is in my sample? I have 10,000 samples. And with a quick back of the envelope calculation, I worked out that it would cost him about 500,000 to have a quantitative assay developed for and run for those large number of samples. But what having talked to him, he actually didn't really want to know how much was in his sample. He wanted to know which samples had any of the compound in. And that's an easier question to answer. And what I recommended to him was that we bought some TLC plates. I taught him how to do TLC. And within a week, he managed to go through his sample set using a simple, cheap, rapid, efficient, and easily scaled assay, and all for the cost of about £50 for the cost of the box of TLC plates. Again, saving money, we can analyze things by HPLC, usually for semi or non-polar analytes. It can be coupled to mass spectrometry with care. Normal detection systems are UV, although you can have evaporative light scattering detection or refractive index if you want. If you
Just
want
a minute to go, Scott. Sorry to interrupt.
A minute?
You. Yeah.
I better hurry up.
Thank you.
Yeah. So mass spectrometry, the complexity depends on the accuracy that you want to use, and usually most people go for triple quadrupoles, which is the pharma standard for quantitative, and it also generally gives you the best linearity, so the widest dynamic range of measurements, because both the bottom concentration and the top concentration may be important to you. So understanding that what the range that you want to measure is also important, because there may be other methods that we can use to concentrate your analyte so we can get you the results that you want. So know as much as possible about your analyte, know what you need to know and what you want from your analytical purpose. Choose a fit-for-purpose method. It'll save you time and money. Ask advice from your analytical chemist. And as a general rule, try to keep as many things as possible constant, including the tubes that you collect blood in, the tubes that you store your samples in, and the number of freeze-thaw cycles that your samples go through. Because all of this can affect your analytical results. And at that point, seeing as my clock now says 25 past, I'll stop sharing and I will take any questions if there are any.
Thank you, Scott, much appreciated. If you've got any questions, please do pop them in the chat. If any of our attendees want to get in touch with you, Scott, is it best to go through your Affinity contact in our booklet or your personal address?
Go through the Affinity if you can, please. So that's scott@affinitybiomarkers.com.
Brilliant, thank you. Jim, would you just be able to pop that in the chat should anyone wish to get in touch with Scott after the meeting? We haven't had any questions come through. If any do, then we'll follow those up online. Thanks for your time, Scott. I know you've got to dash.
Thank you, Becky, and I apologise for missing the next talk. It's one that I would have liked to have seen, but as I explained earlier, unfortunately, I'm double booked. Good luck,
Not
Penny.
a problem. We'll share the recording with you, Scott. You won't miss anything.
Thank you.
Thank you very much.
Bye-bye.
Our next speaker is Penny Cristinacce. She's a research fellow with expertise in quantitative MRI, and she's part of the Quantitative Biomedical Imaging Lab at the University of Manchester. She's also part of NCITA, and she's going to talk to us about that today. Thank you, Penny. The floor's yours.
Thank you, Becky. Let's share my screen. Okay, hopefully everybody can see that now. Hi, so yes, my name's Penny Cristinacce and I am a research fellow here at the University of Manchester. And I'm going to talk to you today about a grant that I'm involved in called NCITA, and that's the National Cancer Imaging Translational Accelerator. So I'm going to give you a flavour of who NCITA are, where the grant originated from. I'm going to tell you why we need NCITA and how it is related to the translational pathway. I'm going to tell you what NCITA does, what we're trying to achieve, and how we're trying to move imaging biomarkers along the translational pathway, and then a little bit about how we can help you here as the Translational Manchester community. So if you can see the map on the right, you can see that the NCITA grant has been awarded to many universities all around the UK, from Glasgow down to sites in London. And the University of Manchester is one of the partners in this grant. And it's a CR UK funded initiative to create a national imaging network for the standardisation and clinical translation of cancer imaging biomarkers. And it runs on the back of a previously funded initiative by CR UK and EPSERG from 2008, the Cancer Imaging Centres. And at that point, the University of Manchester collaborated with the University of Cambridge in order to develop a range of novel imaging biomarkers that now are in a position where we're hoping to push them further along the clinical path, towards clinical application along the translational pathway. As I mentioned, it's a multi-institutional consortium, but it's also cross-institutional. There are 3 cross-institutional units who are working together in synergy to try and create an integrated pipeline that will assist in translating cancer imaging biomarkers so they can be used by the NHS in clinical application and used in clinical trials by pharma. So what we're hoping to create is more quality assured cancer imaging biomarkers that can be used to help patients with cancer. So I've stolen the Translation Manchester lovely schematic to really give you a flavour of where we think NCITA sits along the translational pathway. So you can see that much of the work that is done at universities in research medical imaging really sits on the translatable research end of the pathway. I've been involved for many years in studies that are developing novel methods of acquisition and novel methods of analysis, finding out using imaging in order to really understand and get information about mechanism and metabolism and structure and function. But these imaging biomarkers to be used for clinical application really need to be pushed further along this pathway. And what we are hoping to achieve in the NCITA grant is to really bridge that gap and produce an integrated pipeline that makes it as easy as possible for these methods that are being developed and have been developed for many years in the research communities to be moved towards clinical application. So where are we now and where would we like to be? So the current standard of care for cancer imaging is typically anatomical MRI. So that's used in the TNM staging of cancer, as well as things like the RESIST protocols, and also glucose-based PET. Particularly MRI is often qualitative in terms of its the imaging that's performed, and what that means is that the images that are created from MRI scanners are of arbitrary contrast. So we're looking for difference in contrast, and we're not particularly bothered about what the numbers and those images actually mean. And that is because we, the radiologists, can look at those images and use their subjective expert opinion in order to decide whether or not the diagnosis of cancer and what the treatment pathway should be. But what this means is there's quite often variable acquisition between different sites. Different scanners don't necessarily have to be acquiring data in exactly the same way. However, we have a whole range of tools that have been developed over the years that give us a lot more insight rather than just where is the tumour and how big is it, but we can get information about the tissue microstructure. We can use PET tracers, for instance, to look at metabolism, and we can also look at function using an array of different medical imaging methods. And these methods are quite often more quantitative in their nature, that meaning that we can get physiologically and physically meaningful numbers and importantly their errors from the images that we are creating. And this isn't to say that we don't need this subjective expert opinion. We still do. Much of the standard of care imaging is still very important, but having these other tools at our disposal means that we could improve risk management. We could improve the access to predictive biomarkers, which could help determine which treatment pathway would be suitable for patients as they come through the system. But we need more consistent acquisition if we were to do this, and this is for two reasons. For the quantitative imaging biomarkers to be calculated, we require more consistent acquisition, but we also need to produce multi-center trials of these imaging biomarkers to test their validity, to test the repeatability and reproducibility of the imaging biomarkers, so we can hopefully move them into clinical application. So that was why we need NCITA, but what does NCITA do? So the grant is hoping to provide an integrated infrastructure to ease that translation of cancer imaging biomarkers towards clinical application and use in the NHS. but it also allows access to a range of world-class medical imaging technologies, both particularly in MRI and PET, but we are open to discussion about other methodologies as well. And many of these were developed during the previous initiative of the Cancer Imaging Centres. So we're aiming to support multi-centre cancer imaging biomarker studies and clinical trials, and really bridge that translational gap that I mentioned by providing biological, technical, clinical validation of cancer imaging biomarkers, and also looking at the health economic benefits. And so in the end, what we want to develop are a range of quality assured imaging protocols that can then be used by the NHS and pharma in future. So this is our setup. So we have a governance group that is made up from members of the different institutions involved in the NCITA grant, and we have 3 units. We have the QA/QC unit, the clinical trials unit, and the repository unit. And I'll talk about those in a little bit more detail on the next slide. But we also have a number of groups. So the engagement group is involved in working with the different stakeholders involved in clinical translation of biomarkers. So we're thinking NHS, pharma, patients and service providers, and also letting people know what we're doing. So the news and the events and press releases that are involved. The contracts management group is working to facilitate contracts between the different partners and any external partners, and really make it as easy as possible for multi-centre clinical trials to run smoothly. And the training group is putting on workshops and conferences in order to really train that next generation of cancer imaging scientists, so we can really move forward with this. So the 3 units, as I say, are the clinical trials unit, and very much like any other clinical trials unit, they're there to support the trial design and statistical support, but really with a focus on supporting and coordinating imaging studies, which is what makes this more novel, and they can support both single and multi-centre trial delivery. The QA/QC unit, which is what I'm a member of, is hoping to provide validation and standardization and QA/QC of cancer imaging biomarkers, and we're going to do this in a number of ways by evaluating sites and qualifying sites for inclusion in the trials, for assessing the quality of the data that is coming from the trials once they are active, and establishing these quality assured imaging protocols for use in future studies. The repository unit is using the XNAP platform in order to securely store and share the data from research studies and trials, and trying to make it as easy as possible to access data processing and analysis tools. They're also working on an array of AI and machine learning tools, which hope to lead to earlier diagnosis and more personalised medical approaches to using patient data. In addition to the infrastructure, we also have a number of Exemplar projects, and the reason for those are twofold. So one is to test the effectiveness of the infrastructure that we're creating, and the second reason is for those imaging biomarkers that have been developed to benefit from that infrastructure and to help them accelerate along the translational pathway and validate the biomarkers as we go along. You can see there's a number of Exemplar projects at each of the different sites in both PET and MRI, and just pick out the one from Manchester. We're going to be doing a study alongside UCL where we're using the oxygen enhanced MRI technique in order to look at adaptive radiotherapy in lung cancer. And that's a lot of work that's been carrying on, been happening at Manchester for many years now, and building upon that. As I mentioned, engagement is really important to what we're trying to achieve here with the NCITA grant. So engagement with different stakeholders, as you can see from the figure on the right. So working in collaboration between clinical scientists and trying to establish some consensus about how we can get imaging biomarkers certified and adopted for clinical practice. We're also working with Cancer Research UK commercial partnerships in order to ensure that any of the discoveries that are made during this grant are made available to patients with cancer as soon as possible. So lastly, how can we help you here in Manchester? So there's many institutions around both academic and clinical around the Greater Manchester area, and we're happy to work with both researchers and clinicians that have imaging projects that could need some infrastructure support in order to move them further along that pathway towards clinical application. So you can get in touch with us and get your study adopted by NCITA using 1, 2, 3 of the different units in order to support that, providing you meet some certain criteria. As well, we are developing an MRI core lab. There has been a PET core lab down at KCL in London for many years now, but there's not the same functionality for MRI. And as part of that, a lot of that work is being carried out here in Manchester and builds on work that we've been doing in the Quibi lab for many years now. So much of this is externally facing towards the wider community, but we're learning things here in Manchester that we can reflect internally back to researchers more locally and to people in mass. So if your study is not suitable, for instance, for adoption by NCITA, we may still be able to help on a more local level at the Quibi lab. So that just goes for me to say, if you would like a bit more information about this, you're more than welcome to go to the NCITA website, or you can follow us on Twitter. If you're interested in adoption of an imaging study by NCITA, please contact ncita.general at the UCL address. And if you have any questions, of course, you can ask me now, or you can get in touch on my email address, which is Penny Cristinacce at Manchester. Thank you very much for listening. Stop sharing my screen now. If you've got any questions, I'd be more than happy to answer them.
That's great. Thank you, Penny. We have had a question in the chat around NCITA and Translation Manchester. Have you any plans to work with the Pancaste Institute or other partners through the Pancaste Institute? So I can say on behalf of Translation Manchester that we have worked very closely with the Pancaste and we're part of the operations group. So we're joined up there. Are you familiar with the Pancaste? It is relatively new, Penny.
I think I may have heard of it, but I'm not particularly familiar with it. So I'd be very interested to hear a bit more about it and how I feel that might be a collaboration that would be something that would be very useful.
Yeah, absolutely. So the Pancaste is set up to really bridge across digital and advanced materials in health. So it really sits between FBMH and FSC. So we can share some more information with you on that as well, and the various partners that are involved with Pancaste. But yeah, it fits nicely between the science and engineering and the biomedicine and health.
Excellent. Yes, I'll definitely look that up. But yeah, please get in touch if you want to chat about that in a bit more detail.
Any other questions, please do post them in the chat. We've got a few minutes left. So Jim, would you mind just launching the poll? This is just some feedback for us. As you know, this is the first of our TMRN seminar series. So we just want a bit of feedback from you. Make sure that we're delivering something that's useful. And like I said, any other questions, please post them in the chat, or you can always contact our speakers outside of the meeting. They've provided some details for you. So I think as we've got a few minutes left, I'll do a bit more of a plug on some of the Translation Manchester upcoming events and the future seminar series as well. As you know, we've got planned for the end of March. Just bear with me. If any questions do come up, Jim or Ali, for Penny, then please do interrupt me. So this is the Innovator Training scheme that Ali mentioned at the very beginning. We've been working on this with the BRC This will launch at the end of March, 25th of March. You can follow the link that Jim's already put into the chat if you want to sign up for the launch event, and you can see here the programme for that event. This training is really about getting researchers to think about how they can innovate and think with an entrepreneurial mindset about how to push their research towards the clinic for patient benefit in an effort to commit, and particularly around the idea of commercialising and working with industry partners. All of the Eventbrite links for this programme, which you can see on the right hand side, are now live. So if you follow the link, as I said, that Jim's put in the chat, then you'll be able to register for those events that are all lined up between now and November. Ali mentioned early career researchers competition. This will be an open call in the next couple of months. It's all aimed at supporting the acquisition of translational research skills and progression of translational research. The remit is going to be relatively broad, so training, visits to collaborators, whether that be academic or industry, sharing best practice, and secondments will all be within the remit. Our informatics training scheme, that call will go out again in the summer. So we've ran this the last couple of years. And it will run again this year. It's an opportunity for researchers of all career stages to get an opportunity to gain some new informatics training that they can apply directly to their translational research. And it covers units within existing master's courses at the university in areas such as bioinformatics, programming, stats. So another one to look out for in the coming months. And then finally, our flagship funding schemes, the access to expertise and the projects for translation. These will launch again in the summer with the view of the funding being released in November time. A2E, access to expertise, will pay essentially for the salary or consultancy cost for you to bring an expert into your group, whether that be something like a research nurse for sample collection or a bioinformatician or biostatistician to analyse a large data set. And then projects for translation, this is really about seeing your work progress along our translational pathway. And we, again, cover all stages of that pathway. We will fund work such as proof of concept, but also patient-centred research and collaborations with industry. So I think that brings us up for time. Thank you all for joining us. If you have any questions for us or any of the speakers, please do get in touch with them outside of this meeting. And we look forward to seeing you again at the next session.